Hi, I'm Rebecca Xiong, and together with my colleague, Marcus Starloff, we run the MIT Startup Exchange, which I'm here to tell you more about. Over the last day and a half, you heard a lot of great talks. I've talked to some of you. Many of you found it enjoyable. Hopefully it sparked some ideas for your own work. Now, another part of what makes MIT really awesome is the dissemination of the innovation. So taking these ideas that you hear about, this research, and encouraging the MIT community to quickly commercialize them into startups to more quickly solve real-world problems, the problems you might all face. I myself am a good example. I did my PhD research right here in the MIT Media Lab, and afterwards co-founded a couple of startups with my lab mate. And now I'm back at MIT to help other startups make that connection. The MIT Startup Exchange was founded 4 years ago based on input from ILP companies like yourselves to better connect the MIT startups, 1,700 of them, with industry. These startups come from all across the campus and either license MIT technology or is founded by an MIT alum, faculty member, staff, or student. We know these startups very well, and we also know our member companies like yourselves very well. This enables us to make more high-quality introductions. So every year, we make 600 plus introductions. It's really a great member benefit for you to take advantage of. As you can see, these startups cover a wide range of technologies. We have 60 in AI and ML, another 70 in big data analytics, 50 in advanced manufacturing, and hundreds in life sciences and healthcare. There's probably something here that could be relevant for your business. Here are some of our success stories of partnerships between our startups and the member companies. Arundo is an advanced analytics company, and they've partnered up with ABB to produce the first cloud-based virtual multi-phase flow meter. Now it's integrated into ABB's offering for their customers. And Tulip, which makes ILP-enabled training apps, is partnering up with Merck to help train its operator in complex manufacturing. These are just some of the partnerships that we can mention. There are many, many more. So how can you best engage with these startups? Great question. One way is by attending events like these. So soon we will feature 9 of our startups, and they each give a quick talk, just a few minutes long, to really give you a flavor of what they do. Afterwards, it would be followed by a lunch exhibit where you can talk in more depth with these startups about your own unique challenges. I'll highlight here some of the upcoming events. We have our MIT Startup Showcase event in spring, in March, at Silicon Valley. We also have our Quest for Intelligence conference, a whole conference dedicated to AI and machine learning, coming up in April. And in the end of May, we have our Startup Ecosystem conference. It's our largest startup conference. We also have the STEX25 Accelerator. These are from the 1,700 startups. We picked up 25 that are most ready for introductions with industry. So these are the ones that have proven themselves in early use cases and are really ready for rapid growth. So we help them make additional intros with our companies. Here's a list of current STEX25. We rotate them every quarter. And some of the startups you hear about come from this list. But what if you have a challenge that's not addressed by these STEX25 startups or by those we feature at events? We have another way. It's called an opportunity. So here companies can post the challenges they have And that challenge will be sent out to all 1,700 of our startups so they can directly apply to work with you. You see here a couple examples of postings by our member companies for geointelligence solutions or for robotics. So now we have the startups come on stage to talk. Here at MIT, we like to give homework. So here's our homework to you. Please think about, as you hear these startups talk, ways to apply these innovation with your work. And if they apply, then come talk to them at the lunchtime exhibit. Or if there are challenges you're thinking about at work that's not covered, you can talk to your ILP contact or get in touch with me, or come find me during lunch as well. So these talks will go one after the other with no questions, so please hold them until lunchtime. Without further ado, here are the talks. Great. Yeah. Should we go in order?
I ought to be anyway. A few slides back. Great. Awesome. All right. All right. So, hi everyone. My name is Charles Fracke. I'm the CEO and founder of BioBright. BioBright is a company out of the MIT Media Lab and Harvard Medical School. And for the last 2.5 years, we've been deploying kind of high-impact digital technologies with biotech, pharma, and we're now expanding into healthcare as well. What I want to talk to you about today is about how to use digital technology to create actionable and meaningful results in the field of life sciences. From our experience, there are 3 major problems that you're facing as a large company to actually use AI and digital technologies in your company. The first one is collecting the data in the first place. Don't make the assumption that the data is available. This is one of the major issues that we see. Second, once you get that data, okay, you need to clean it, you need to use it, and actually make it into a format that can be reused. And once you've done that, you've got to analyze that data and make it actionable for the end user. There's no point in sitting on a ton of data that scientists or business intelligence people cannot actually make action upon. So here's a picture of what it would look like today, what you're up against. First, you need to collect that data. Let's say that this data comes from instruments. Some of them will be connected to the network, some of them won't. And then you need to centralize all that information locally at your site. Let's say that it's here in Cambridge. And so you need to maintain all those hard drives, and there's a lot of overhead maintaining that information. Then, if you have multiple sites, and in particular, if you want to make your data available across your organization, you need to put it somewhere that is accessible to everybody. And often that means putting it in the cloud. Then once you've done that, you've got to analyze it, extract, transform, load it in a way that actually brings those insights out from this raw data. And finally, you can present it to the end user and the person that's going to make action on this data. BioBright does all of that basically in one place for you. Oop. This is the slide I want to show. There we go. BioBright does that for you and all in one place. So it collects it, it puts it in the cloud, it can do ETL, so extract, transform, load, and present that information on dashboards that are meaningful for the end scientist or business intelligence person. So once the data is up in the cloud and you can analyze it, one of the things that we do, obviously, is some machine learning on the information. And one example that we're sharing today is an example with Beckman Coulter, a Danaher company, where we double the precision of a particular instrument that is particularly popular in cell line development. Because we had access to a wealth of information, of metadata, that was just not extracted before. So qualitatively, what does that look like? Here's the system before and after. In this particular case, we're looking for cells of a particular marker. Our system is able to find that with a much, much greater accuracy. Not only that, but we are able to automatically detect broken equipment just from the information that's coming off of the instrument itself. So we're able to identify anomalous behaviors and flag it for review or for fixing. It is not limited to just purely log data or technical data. We can also go into the scientifically relevant data. This is a particular strength of BioBright due to our kind of biological background, is that we're able to automatically analyze and visualize assays at scale once this data comes through our system. So we were able to use this with a European pharmaceutical company and help them find a root out cause of error that had been missed for several years in their workflows. Over the last 2.5 years, we've gotten a lot of press and kind of recognition, but now we are actually deploying at scale. We have a package that is very easy and cheap to deploy, so if you're interested in doing this at your company, just come talk to me outside. Thanks.
Good morning, everyone. My name is Dr. Corey Kidd. I'm the founder and CEO of Catalia Health. I did my PhD here at MIT in the Media Lab from 2001 to 2007 at the intersection of artificial intelligence, psychology, and medicine. Catalia Health builds chronic disease care management programs. And we start with the psychology behind what makes healthcare work with patients. And a lot of that is the human touch. In a lot of healthcare, we spend a lot of time with our patients in the clinic. Now, the challenge in dealing with chronic disease is so much of that happens outside of the clinic, outside of the hospital, outside of the doctor's office, the face-to-face settings. And we spend a lot of time working with pharmaceutical manufacturers right now for patients on specialty pharmaceuticals where there are care management programs that today look like a call center full of nurses that call a patient maybe once or twice a month, which is great for providing a little bit of support, but the actual challenges that come up happen day to day. So starting with the understanding of what works in terms of interaction with patients, we have built artificial intelligence algorithms that can generate conversations on the fly for each patient at that point in time. And so for the same price point that we can have a nurse calling a patient once or twice a month, we can talk to our patients once or twice a day. Now, the way that we deliver this is unique. I've talked a bit about this, so I'm going to skip over some of the challenges here. The way that we deliver this is a bit unique. So we use a robotic interface. So this cute little yellow robot, and I have one in the exhibit area that you can come talk to over lunch, is what we put in front of our patients. And now the rationale behind this, putting this in the home, is not a gimmick, but goes back to that same psychology I was just talking about. You know, the reason that we're all here in the room, as opposed to putting these talks up on a website or, you know, sending out some materials, is, you know, giving, you know, these talks is one thing, and then now the ability and the networking breaks and over lunch to talk to one another face to face really makes a difference versus, you know, getting this information disseminated electronically. And we know what those differences are. When we're face-to-face with someone, we're more engaged, we create a stronger relationship, that relationship lasts for longer, and that's why the clinical setting works so well in healthcare. Now, what I found during my PhD work about 15 years ago is that those differences actually carry over into the world of technology. So again, most of what we build on the technology side at our company is the AI to create conversations, but when we deliver it through an interface like this that can literally look our patients in the eyes while it's talking to them, we get those psychological effects of face-to-face interaction. Now, we use those conversations to talk with our patients on a day-to-day basis. We start with that foundation of building the relationship, and so what we've shown through doing this is we're able to keep patients on therapy for significantly longer. They will use a system like this much longer than delivering the same conversations through a screen-based interface, you know, the phones that we all have in our pockets, or a tablet or a television. Now, this is not the only way that we interact with our patients. We also can text through apps and other things, but we use this as really the key way to generate that relationship that's ongoing. And because we're talking to our patients so frequently, we now have a lot more data about what's going on. So that's very useful when we're creating new drug programs and clinical trials. We're starting to do some work there to be able to track patients. But for patients on therapy in the real world, we're also able then to intervene if and when necessary. So this is live with patients. Our first product that's out was launched earlier this year. One of our first customers was Kaiser Permanente. That's our heart failure product. We are working with the American Heart Association on that. Before the end of the year, we're also launching products in rheumatoid arthritis and late-stage kidney cancer. So we're working across a number of different disease states. And we're looking for partners who are seeking solutions that can really interact with patients on a frequent basis over an extended period of time. So I look forward with talking with you over lunch about that. Thank you.
Hi, I'm Steve Harrison. I'm the Chief Scientific Officer at Engine Biosciences. Engine is a drug discovery company that was founded out of MIT from the labs of Jim Collins and Tim Liu, also with Hu Li out of the Mayo Clinic and Prashant Mali at UCSD. And what we do is we're a drug discovery company. We identify targets and biomarkers, and we're using machine learning tools to interrogate large amounts of human clinical data, but really importantly, to understand the underlying networks of genes that are different in disease cells versus normal cells. And then, importantly, we test the hypotheses that we get from the computational work in the lab in high-throughput functional genomic approaches using, for example, CRISPR-Cas to test what happens when we manipulate the genes that we've identified. So as you recognize, the problem here is that there are 20,000 genes in the human genome. They're all interacting. Those interactions can be different in disease cells versus in normal cells. But if we can get some insights into that, it'll help us to understand the multiple factors that are involved in causing disease. It'll tell us what are the unique vulnerabilities in disease cells and help us to identify biomarkers, not only which patients will respond to a therapy, but when they respond to a therapy. So this is the workflow that we do. We take clinical data, both public data as well as proprietary data. We're a company that's based in Asia. We have access to a lot of proprietary clinical data in China. We are using our machine learning algorithms to understand the differences in the networks of genes in the disease cells. And then really importantly, we don't believe that the computational work alone is going to identify the key biomarkers and the key targets. So we are then applying, what we're doing is identifying hundreds or thousands of possible targets, which we then validate in the lab in human cells. And those, as we heard earlier today, could be iPSC-derived cells or from patients, or they could also be, for example, tumor cell lines. And they use a technology known as Combigem, developed out at Tim Lu's lab, that allows us to do high-throughput manipulation. That will identify targets that are of interest. We can develop those within Engine. We have the capabilities to do drug discovery. But really importantly, the data that's coming out of those screens, whether we were right in our hypotheses or not, feeds back into the algorithms and tells us in the future to train those algorithms to make them function better. We're focused in cancer as well as in neurodegenerative diseases. We are looking at synthetic lethals in a number of cancers, starting in an Asia prevalent disease, because we're based in Singapore, and ultimately looking at other diseases. We're looking at the drivers for successful autologous T-cell transplantation. What are the pathways that one would want to augment or diminish to have success? And we're also looking in neurodegeneration. In fact, we've got some interesting therapeutic candidates in ALS. We've also been working with industry, for example, with Procter & Gamble, to look for some factors that are helping increase skin cell viability. So we're very interested in collaborations with leading laboratories to develop new network-based algorithms and to enhance functional genomics platform, and we're open to suggestions for additional applications and potential industry partnerships. So I'm Steve Harrison, and please come see me after the presentations. Thank you.
At Interpretable AI, we turn data into trusted action. I'm Daisy Zhuo, co-founding partner, together with MIT professor Dimitris Bertsimas and Dr. Jack Dunn. We all know the future of medicine is artificial intelligence, but the reality is that AI must work together with humans for it to solve the toughest problems, like drug discovery and curing cancer. Many people are realizing this now and are calling for action. The FDA chief said they're not comfortable with the approval of using AI without the level of confidence in what it's doing. A recent article in Harvard Business Review says that companies will not be able to take advantage of the huge potential of AI if employees don't trust it. Another article in Nature this month said that model performance will not matter if the algorithm design neglects the need for trust, and because of that, opening up algorithms for interpretability is a necessary first step. So collectively, for AI to thrive, it must explain itself so that humans can understand, validate, and most importantly, trust what the system is doing. If we do not understand the logic behind these systems, they will break down. At Interpretable AI, we deliver interpretable solutions to your toughest problems. Our solutions leverage our proprietary software modules, each based on years of AI research at MIT. With these software modules serving as the building blocks, our solutions are able to deliver state-of-the-art performance while still remaining fully transparent and understandable to everyone at your organization. Our technology spans the full spectrum of data analytics, from data cleaning to predictions to data-driven decision-making. Our technology has applications in many areas, but in particular, we have an established track record of delivering impact in medicine with interpretability. So some of you might have seen this from an earlier talk. The top surgeons at MGH trust the predictions that come out of our surgical risk calculator because they can go into the system and validate this, the logic behind the system. In the case of oncology, we help patients and doctors to better understand their prognosis and therefore come up with more informed treatment decisions by using a cancer mortality calculator that we developed with Dana-Farber. In drug discovery, we used our software modules to identify novel targets of existing small molecule drugs so that to open up opportunities to design these highly specific prospective trials. And finally, even with failed trials, we analyzed their data and we were able to identify an interpretable subgroup of exceptional responders so that, again, opening up the possibility to revive these drugs, which unfortunately failed after so much investment. So we're looking for a number of pilot partners that need trusted solutions to their hardest problems. For example, we can help with drug discovery, with clinical trial planning, and with post-trial analytics, as well as any other problems that you think can really get some value from interpretability. Come talk to us if you want to improve the trust in your data-driven decision-making.
Good afternoon. My name is Matt Osman. I'm the CEO and co-founder of Legit. We're a natural language processing company originally out of CSAIL that uses NLP to increase efficiency in the R&D process for healthcare and life sciences companies. So the mission of the company is to move away from what we call kind of point-in-time access of technical literature. So people review technical literature for a lot of different reasons, finding external alliances, determining the viability of a new strategy in device or drug formulation, but we want to make it continuous. We want to continuously monitor and acquire and provide to R&D teams all the information that would be required to create novel, valuable, and clinically effective devices and formulations. And that includes both internal documentation and also voice of customer data and external data as well. So the problem, quite simply, is this. What you'll see on the right is the growth in scientific papers and citations. Scientific output doubles in terms of literature every 9 years, and in healthcare, it's actually shorter than that. Not only that, the data sources for internal R&D and external R&D efforts are too numerous for human review, apart from maybe on a point-in-time analysis. They're also poorly tagged and ambiguous. So I'll give you 2 examples. The laser, it's a pretty standard technology. The word laser does not appear in the patent for the laser. ERBB2 is also known as HER2. You have smart people in the technical literature describing the same thing using completely different terminology all the time. They've done it since science began. Not only that, but talking to customers, talking to people who are actually going to be using your devices or prescribing your formulations is difficult and expensive to acquire. So the technology we've built to solve this problem is an NLP system which can quantitatively measure how similar two pieces of technical text are, even when they don't share keywords. So we know the optical maser refers to laser. We know the ERBB2 is HER2. We do that by creating a 500,000 dimensional concept space, which is then dynamically trained on user interactions and technical literature. So what this allows you to do is to build a conceptual RSS feed, essentially, that you can point at almost an unlimited amount of text data sources for particular use cases. So this is how we see it used. We can build predictive models to predict potential defects in devices or formulations by comparing R&D text data against FDA defect and adverse event data coming from manufacturers and surgeons, and that's updated daily. So we can tell you that your surgical robotic arm is conceptually very similar to something from a competitor that killed someone in a hospital in Cincinnati yesterday. We can do the same thing to predict what surgeons and clinicians want by passporting your voice of customer data, which is in a CRM, or allowing you to acquire it through us. So we have a deal with the Mercy Health system such that we can find doctors for you to talk to that are similar to the devices that you're working on to get real-time feedback before you go down a rabbit hole over 9 months. We can identify product differentiation by comparing R&D text data against internal documentation, patents, PubMed, NIH grants to tell you, well, the thing that you're working on is 92% similar to something you tried 7 years ago and it failed. Why is it going to be different this time? And because we can do that cross-matching, we can eliminate R&D wastage. So we can identify if you've done this before or if it's being done across silos within a company that is distributed. We can also identify strategic opportunities outside of the company. So you can point this data feed at academic research, new FDA registrations, financing data, and individual researchers that you particularly want to track. The users that we work with typically are research engineers, scientists and their managers, product development teams within healthcare, and people on S&E and external alliances teams, as well as BD. These are some of our partners. As I mentioned, Mercy Health are providing access to their clinicians and doctors to some of our early-stage medical device companies. If you'd like to discuss how we might be able to work with your company, please come and see me at lunch. I'll be the sleep-deprived British person in the corner. Thanks very much.
Great. Hi guys, my name is Susan Conover. I'm co-founder and CEO of LumenDX, and we identify skin conditions instantly using, does anyone want to guess? Yeah, AI, you got it. So we have an amazing team. My background's actually in mechanical engineering. I worked in management consulting, and then I came back to school at MIT and got a degree in engineering management. That's where I met my co-founder, Jim Barkley. We also have 2 amazing dermatologists on the team and phenomenal technical business and marketing experts to grow this company. So I don't know how familiar you guys are with skin disease, but 1 in 4 Americans seeks help from a physician for skin disease each year. And 2/3 of these cases are actually dealt with in primary care. Crazily enough, 50% of these cases are misdiagnosed. So both providers and patients struggle to visually assess skin disease and understand what to do next. And on top of this, dermatologists, there's a mismatch in supply and demand by a factor of 4 times. So you can't just refer out all cases. So what we've built is a system where you take a picture of a skin disease problem, and then using our cutting-edge deep learning trained on thousands of diagnosed images, we instantly identify the most visually similar diseases to the picture you took, and then connect patients onward to physicians. We're first going to patients at home, and then going to physicians in the clinic. So we launched our beta in August, and it was fairly successful, I must say. We trained our system on 8,000 images of actually genital skin diseases to help patients who are concerned about what's going on in that area, don't want to talk to friends or family members, but understand what's going on at home, and then get connected onward to physicians. And our strategy is twofold on building our data asset, because we all know AI, in AI, data is king. Right? And so we're working with physicians who are sharing their diagnosed image data sets with us, in addition to sourcing images directly from patients at home who have the most to benefit and are the most concerned about skin disease. So as I mentioned, we trained our model on 8,000 images. We recently actually brought in an additional data set to double our images. So now we're up to 16,000 specifically on genital. And we launched our beta with greater than 10% click-through rate. We're rolling out a new version in about 2 weeks. And there's a question that I always get, which is, how much data do you need in order to get to whatever accuracy you're shooting for? So we built a model to show the different 3 axes that we think are the most important: data quality, data variety, and data volume, right? And so to go into primary care, we've predicted we need about 370,000 images across, 20 different body parts, multiple skin colors, multiple diseases, in order to be able to really help a primary care doc. So basically, I'm here today sharing our progress, and then we're also, of course, looking for additional data sets, so physicians to work with in dermatology, but also infectious disease, OBGYN, urology. And then if you're interested in a partnership and there's something I haven't presented today that you have something in mind, we're, of course, happy to discuss that. Thank you so much.
Hi, everyone. I'm Aditya, the co-founder and CTO of PathAI. And a couple of years ago, I got my PhD in computer vision and deep learning from CSAIL. And essentially, the problem that we've been working on is pathology. And basically, pathology is the area associated with disease diagnosis, right? So essentially, a lot of the ground truth for many of the skin conditions that were previously mentioned come from pathology. And pathology is kind of, after it seems like something is wrong, you go in, you talk to a pathologist, and they do a biopsy, and what you get is some sort of diagnosis. And oftentimes, pathology is seen as medicine's ground truth, which guides all downstream decisions for a patient. Unfortunately, there's a lot of disagreement amongst pathologists, and there's a lot of error. So this graph right here shows it's a pretty simple but scary experiment. So they took a bunch of patient samples, and for each sample they actually showed it to about 30-odd pathologists independently. And one would hope that a patient either has cancer or they don't have cancer, especially if this is the ground truth that we're presenting to patients. But what each line shows here is the each line here, vertical line, corresponds to a patient, while each color corresponds to a diagnosis. So what you'll notice is that there's actually, for some of the patients, it's almost as if it's like a uniform distribution of diagnosis, as though somebody goes into a room, tosses a coin, and tells the patient whether or not they have cancer. So this is kind of the state of the art. And as you can imagine, how can we solve it? It's using AI. So what we can do instead is essentially you can imagine a whole slide image such as this one. So this is a typical pathology image. It's a humongous image, so it's something on the order of like 100,000 by 100,000 pixels. You can zoom in and you can look at the nuclei of each cell, and this is the level at which diagnosis is typically done. So using an AI system, what you can do is you can start. I guess that was supposed to be an animation, but you can start quantifying, different aspects of the tissue. So what you see in red is, the regions identified as cancer. What you see in green are regions identified as stroma, and in black, necrosis. And you can go beyond and start identifying different cells as well. And so now this is now quantifying how that system does. In a competition, this happened somewhere back in 2016, where the goal was to identify metastatic breast cancer. In that competition, it's like an ImageNet style competition where you're given a training set and you have a test set and you're supposed to, provide a diagnosis on the test set. So there were 2 ways pathologists were evaluated. The first was in a competition setting. So they were told, hey, take as much time as you need. You're going to be competing against a machine. Do your best. And in that case, the pathologist had an error rate of about 3.5%. Whereas if you switch to a clinical setting where a pathologist is looking at something on the order of 200 to 300 slides every day, so they have a limited amount of time to go through each case, the error rate actually increased to 13 to 26%. While the error rate of our fully automated system was at something of under 1%, at about 0.65%. And the hope is that by combining our system with a pathologist, we can bring this error rate down to 0. So, what we've been doing beyond diagnosis is actually working with, pharmaceutical companies and, other drug development companies to identify the best set of responders based on, patient pathology samples from clinical trials. So far, we have a number of partnerships with Philips, the Bill and Melinda Gates Foundation, Bristol Myers Squibb, Novartis, and a number of other pharmaceutical companies, and have worked on a large variety of cancers listed here, and I think the list is actually longer than that. And we're looking to partner with other more pharmaceutical companies looking to use pathology more effectively. So in summary, PathAI is building the world's leading computational pathology company, and we use AI to both improve diagnosis as well as the power of prediction to accelerate drug development for cancer and other serious medical conditions. And I look forward to chatting with folks in the next few sessions.
Hi, I'm Leila, founder and CEO at ReviveMed. At ReviveMed, we use AI and metabolomics for drug discovery. The technology is based on a work I did during my PhD at MIT at Ernest Frankel Lab, who gave an awesome talk earlier this morning. You guys all know about DNA, right? DNA will transcribe to RNA, then RNA will translate to proteins. These proteins then interact with tens of thousands of small molecules or metabolites in human body. These molecules are like sugar, cholesterol, all the fats. These interactions lead to what to observe in clinical phenotype. So metabolites provide the most functional information about disease. However, in the last decade, because of the availability of genomic data and compute power, AI platforms developed to leverage genomics for drug discovery. Now, at ReviveMed, we are able to use metabolomics to solve this puzzle. The reason that metabolomics has been underutilized until now, because they have tremendous diversities in their structure. So to characterize a large set of them for each patient, it requires lots of customized experimentation. And that's why current platform can only detect less than 5% of human metabolites. At MIT, we developed an AI platform to solve this problem. We first created the most comprehensive database about metabolites, their interactions with proteins, and association with diseases. We then start from tissues or blood of patients. We do mass spectrometry. By doing that, we are measuring tens of thousands of these small molecule masses. But the issue is that we don't know what they are, because there could be a 10 metabolite with exact same molecular mass. I have this dummy example. If it's something 180, it could be either glucose, fructose, or galactose. They all have the same molecular mass, but different functions in our body. Current platform, they have to do more experiment to figure this out. What we are doing instead, using our AI platform, we are looking at those masses that are changing in a disease and consider all those possibilities that they could be. And from that big networks that we created, we reached to this optimum network that connect those masses. And then, through this connection, it would tell us, okay, this 180 is glucose, because how it's connected to the rest of the things that are changing. But more importantly, we end up with this optimum network that gives us critical therapeutic insight. That what is changing in a disease? Because some of those proteins can be a therapeutic target. And some of those other metabolites could be an existing drug. We We validated our platform by studying Huntington disease. We were able to find novel disease pathways. We were able to find novel target, existing drugs with new therapeutic effects for Huntington, which we validate them experimentally and publish in Nature Methods and Nature Communications. Right now at ReviveMed, we are creating value for both patients and pharma companies. We have internal programs focused on metabolic diseases, especially on fatty liver disease, because metabolites play a key role in these indications, and there is significant needs for patients. In addition, we have already established collaboration and partnership with pharma companies. We have the best team to unlock the value of metabolomics. We have been an award-winning startup. We are very thankful of support of industry, particularly from Novartis and Bristol Myers Squibb, who has sponsored 2 years of our lab space. And we are looking for more partners. I will be happy to talk with you guys during the lunchtime. Thank you.
Hi, I'm Andrew M. Radin, co-founder of Tuzar. Yesterday, you heard from Andrew A. Radin, our CEO. Yes, we have the same name. No, we are not related. Tuzar is an AI-driven drug discovery company, and what that means for us is that we use our technology internally to identify new drug candidates. We then validate those in animal studies, and then we partner those programs with biopharmaceutical companies to move it forward towards the clinic. I founded this company in 2014 with the other Andrew when I finished my program here at MIT Sloan. The other Andrew was at Stanford. He created the technology initially, and when we launched the company, we were able to raise some initial financing from Andreessen Horowitz's BioFund, which enabled us to run proof of concept studies in several indications, and I'll show an example in just a moment. We had some nice results from those proof of concept studies, which gave us some nice recognition from a variety of organizations, including an opportunity to meet with a delegation from the Obama White House. In addition, we used those results to sign partnerships with several pharmaceutical companies, including our flagship partnership with Santen Pharmaceutical in glaucoma in 2017. We then, after signing several collaborations earlier this year, closed an additional financing round from SoftBank Ventures, which is enabling us to build a portfolio of drug programs, both internally and in partnership with pharmaceutical companies. After we run our software, the first step is to take our drug candidates and test them in animal models of the disease. These rats have been induced with rheumatoid arthritis, and it is supposed to be a video. The rat on the left is on a candidate that we have identified, and we can show the video at the break, but is running around the cage, living a normal life. The rat on the right here is a control animal and is nursing her swollen ankles and not moving around. This candidate that we identified has never been tested in rheumatoid arthritis before and seems to be acting on novel biology. But I think what is more interesting is that normally, to get this kind of result in an animal study, one expects to test 100 candidates and have 1 or 2 that show this kind of efficacy. In this program, we tested 10 candidates, all seemingly acting on independent novel biology, and 3 of them showed statistically significant efficacy across standard endpoints. From deciding to launch this program to having this video was only 4 months. And this is just one example. We've done this in a number of indications. We built our platform as a scalable software platform that enables us to plug and play disease data into the platform and identify new candidates. We have animal results in immunology, oncology, and metabolic indications. We have modeled a number of disease programs or disease models within our platform and launch a new disease into animal studies on a monthly basis, again, internally and in collaboration with biopharmaceutical partners. One example of that partnership is one with Santen Pharmaceutical. As you know, they have more than a 100-year history in ophthalmology, and so for us, they're the best partner to work with in glaucoma. They were looking to fill their pipeline with innovative therapeutic opportunities, and they approached us to help. We're using our technology to identify new candidates in glaucoma. They're using their ophthalmology expertise to run preclinical studies, and they have purchased the exclusive right to develop and commercialize these candidates. So as an AI-driven drug discovery company, we focus on identifying first-in-class therapeutic opportunities and partnering them with folks. And really, our goal is working together with industry to be able to fill the preclinical pipeline much more rapidly. Thank you very much.
We're having lunch next, so if you have questions or want to connect more with the startups, please feel free to do so. And also, I want to mention, just a tip, a lot of times people eat first and then go talk to the startups, so there's like a line. So the tip here is to talk to the startups first. I promise the lunch will still be there. Another thing to mention is we also have a Hacking the Medicine from MIT team there, and they have a lot of experience working with industry to put on events using a very entrepreneurial approach to address industry problems, like with hackathons. So enjoy, and thank you.